Good morning, distinguished guests, ladies and gentlemen. It is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with the introductions of our special guests of honor, allow me to provide a brief overview of the initiative that brings us together today, Instal Africa. Instal Africa, the Innovation Science and Technology for Action Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. Instal Africa was born from a collaboration between the Foresight Institute of Research and Translation, Rwanda, FIRAT Rwanda, and its technical partner, Drosophila UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. Next, we will move on to narrating the profile of our invited guests.
Dr. Rashidatu Abdulazeez is a rising leader in biomedical innovation and biology education in Africa. She is a lecturer and research scientist at Ahmadu Bello University, Zaria, Nigeria, and currently a Royal Society Fellow at the Institute of Ecology and Evolution, University of Edinburgh. A passionate Drosophila biologist, she leverages this powerful model organism to explore ecotoxicology, population genetics, and host pathogen co-evolution. Dr. Rashidatu Abdulazeez holds a PhD in zoology with a specialization in molecular genetics. Her research centers on understanding how environmental stressors, such as toxins and pathogens, impact the genetic and physiological responses of Drosophila melanogaster. By combining molecular techniques with ecological insight, she investigates the dynamics of adaptation, resistance, and co-evolution in host pathogen systems. Her work in ecotoxicology particularly addresses how pollutants influence gene expression and reproductive fitness in Drosophila, offering broader implications for environmental health and toxicology studies in sub-Saharan Africa. In the field of population genetics, Dr. Rashidatu Abdulazeez examines genetic variation and structure within natural populations of Drosophila, which can offer crucial insights into evolutionary processes, local adaptation, and species resilience. Her research on host pathogen interactions further explores how microbial challenges shape immune responses and drive genetic change over time. This line of inquiry not only advances fundamental biology, but also has relevance for understanding vector-borne disease dynamics in public health. She has published widely in international journals, contributing to the global understanding of evolutionary biology and environmental genomics. Her scholarly excellence and dedication have been recognized through several prestigious honors, including the TWAS DFG Cooperation Visit Award and the NIF Research Fellowship Grant, which have supported collaborative research and capacity building. In 2018, she co-founded Droso for Nigeria, a pioneering initiative dedicated to enhancing science education in Nigerian secondary schools through fruit fly research. Her outreach efforts have inspired thousands of students and teachers across several states in Nigeria, promoting local capacity in biomedical research and hands-on inquiry-based learning. Through her work, Dr. Rashidatu Abdulazeez continues to bridge science, education and community, earning her a place among Africa's most impactful young scientists. Her unique integration of world-class research with grassroots scientific outreach exemplifies a model of scientific leadership grounded in both academic excellence and social relevance. So, without further ado, please join me in welcoming Dr. Rashidatu Abdulazeez. Dr. Abdulazeez, the floor is yours.
Hello, everyone. Can you hear me? Okay. Good day, everyone. It is a privilege to speak with you today about a journey which began with a challenge and one which grew into a vision. A story of not just about a fly, but about a movement. The story of hope, resourcefulness, and the power of science to transform education in Nigeria. I will be sharing with you a lived experience that connects education, innovation, and the power of scientific tools to create lasting change. Let's begin with what you actually learned from this talk. It's hoped that at the end of this talk, you will be able to learn about what Drosophila is and how it can be used in research. What Drosophila is and how it can be used in research, from genetics to drug discovery. You will be able to also get simplified lab protocols. You will be able to also get simplified lab protocols. Just a minute. Yeah, okay. Sorry. It's taking a while. So you'll be able to get simplified lab protocols and see how to run experiments with basic resources, and to also hear how Drosophila Nigeria supports students, teachers. I don't know why my slide is not moving as it should. Yeah, supports students, teachers, researchers to connect to a global network. And finally, you will be able to understand how you can build your career pathway. So, a little bit about me. I had my first degree. I don't know why it's not clicking. It's not moving as it should. I'm sorry. So a little bit about me. I had my BSc and MSc in biology from Department of Biological Sciences. Then it was biological sciences, not biology as it is today now, from Ahmed Bello University, Zaria. I had my PGD, that's Postgraduate Diploma in Education, in 2014 from National Teachers Institute, Kaduna, because I love to teach. And I had my PhD in Zoology in 2021 from Department of Zoology, Ahmed Bello University, Zaria. And currently, I'm on my postdoctoral fellowship at the Institute of Ecology and Evolution, University of Edinburgh, where I keep studying Drosophila. So that's a bit about my journey, but it was never just about me. Along the way in 2016, first of all, I started working with Drosophila in 2014 during my MSc, when one of my MSc supervisor, Professor DM Shehu, asked me to work with it, and that was how my journey with Drosophila actually began. So, but along the way in 2016, I realized that the most important thing isn't just what I can do with Drosophila, it's what we can use it for across generation. You know, oftentimes we often treat science as something that is elite, something that's done only in advanced labs. But what if I tell you that true innovation actually starts at the grassroots? And if we can really if we really want to make a lasting progress, we must build generational capacity. So, what is generational capacity? Generational capacity simply means building and passing on knowledge, skills, and expertise to the next generation in order to ensure that there is continuity, sustainability. It also involves building upon each generation's contribution, that is ensuring that there is continuous growth and innovation in the society. So, how can we achieve this? We can achieve generational capacity by first educating students to ensure that they have a strong foundation in critical thinking skills and research methodologies, by empowering teachers, by providing them with the necessary resources and training these teachers to effectively mentor and guide the students, and also by supporting researchers, because over time the trained students become the researchers' expertise and the cycle continues. Another way we can build generational capacity is by cultivating a mentorship culture, which preserves institutional memory and expertise by effectively passing down knowledge and skills across generations. In a statement by Professor Chamberlain during his talk at the BB STEM, that was last 2 weeks, I think. Yeah, he said something that was quite catchy. He said, "To be the first is not something to be proud of, but it's in ensuring that one is not the last." So, encouraging experienced researchers to mentor newcomers would foster a supportive environment that promised professional growth. Well, not just professional though. Then another way to build capacity is by retaining scientific talent. Now, we can achieve this. It's not just by producing the talent. You've trained the teachers, you've empowered the researchers, you've also trained students and the like, but it's about building an ecosystem. It's about building an environment, building the condition which encourages people to stay, to thrive, to grow, contribute, and recycle those talents across generations. Now, whether they are working locally or remotely, that way, once what we once looked as talent loss can actually become generational gain. So, if we want to grow, if we want to retain, grow talent across generations, we need tools. We need tools that are affordable, that are accessible, and that they are powerful enough to support both learning and discovery. And that's where this tiny fruit fly comes in. Drosophila melanogaster, mostly. That's the fruit fly. Now, it may look insignificant to some of us for now, but it has transformed science around the world, and it might just be the best kept secret for building scientific capacity here in Nigeria. So, ladies and gentlemen, let me introduce you to Drosophila. Okay, sorry, just a minute. Yeah, so this is the fly, the small fly, big impact. So morphologically, these flies can be distinguished. The males and females can be distinguished. The adults can be distinguished. They have also morphological life cycle. You can be able to tell the embryo or the egg from the larva, the pupa, and also the adult. So they have a complete life cycle stages, complete metamorphosis. And you can also distinguish them just like how humans vary. These flies also varies, and you can also differentiate them from the different types. We have the wild type, we have the mutants, we have the transgenic flies. Now, also these flies, we find them on mostly on our rotten vegetables, on our rotten fruits, in our kitchens. By the way, they are not harmful. They are not vectors, so don't be afraid. So we can have them in the wild, outside on rotten fruits, vegetables, or we can have them in the lab where we prepare some kind of meal for them, and then they are able to survive and replicate effectively. And also, based on their life cycle, these flies have just within 7 to 10 days, especially I'm talking about Drosophila melanogaster. Remember I said we have different types, just like how human differs. We have different types, different strains. So, but based on Drosophila melanogaster, it has about just 7 to 10 days. It takes about 7 to 10 days for these flies to mature from being an embryo to an adult under an optimum temperature of 25 degrees centigrade. So, and finally, they have 4 pairs of chromosome, unlike we humans that have 36 pairs of chromosome. Now, watch out. That is one of the things that we'll come to talk about later. So, what can this small organism actually do? It turns out it can actually do a lot. From identifying genes that regulate both development and diseases to rapidly and affordably testing compounds in drug discovery, thereby accelerating the development of drugs and effective treatments. The flies can also be used to understand the intricacies of brain function and behavior, thereby can offer insights on several brain neurological disorders such as Alzheimer's, Parkinson's, and the like. We also use the fruit fly to efficiently access chemical safety. That is ensuring that new compounds are safe for further development, which is also one of the field I'm into. That's the toxicology, but I'm into ecotoxicology. That's toxicology, the toxins from the environment. And the field of microbiology is not left out. It's also used to explore host pathogen interactions, providing vital information for combating infectious diseases. And of course, in terms of population and evolution genetics, we use the fruit fly, among other things, to understand the dynamics that shape biological diversity and adaptation, which is my field. So, fun fact, do you know that 60% of our genes and that of the fruit fly are exactly the same, like exactly the same, no difference, right? And also that we can use this fruit fly to study about 75% known human diseases using this fruit fly. That like diseases like cancer, epilepsy, diabetes, and the like. Impressive, right? Yeah, but it's not just about what the fly can do, it's about why it does it so well. Let's explore why scientists around the world and increasingly in Africa are choosing Drosophila. First of all, it has rapid life cycle. Remember, I said it has just within 7 to 10 days, you have your flies, you have your adult flies, and powerful genetics. Those 4 pairs of chromosome I told you earlier, they are one of the key things that make Drosophila to be thriving. You see, for instance, as a teacher in biology, I'm mostly talking about biology. As a teacher in biology, you want students to have a practical experience of one of the topics, like take for instance either reproduction or life cycle. You can get the students to get the flies from the wild, just from their mother's kitchen. They can put it in different bottles, just containers in the classroom, and before the next practical session of your class, these students would have been able to experience the 4 developmental stages. So how practical can that be? Now, also, this fruit fly has been able to has gotten like 6 Nobel Prizes in medicine and physiology. That is, using this fruit fly has gained about 6 Nobel Prizes in physiology and medicine. The last, but not, of course, the least, it will keep coming. It was in 2017 on the discovery of circadian rhythm, the mechanism behind circadian rhythm, such as how we're able to sleep, why we're able to sleep at a particular time, how we're able to wake up at a particular time. All that was discovered using Drosophila. And also, another thing that makes this powerful is the fact that it is cost effective and it just requires a low amount of money to maintain this. For like the example I gave for a teacher wanting to teach life cycle to the students, it doesn't cost anything. They are all reusable things. You get used water bottles from from homes, you get some rotten vegetables also from homes, and then you just keep them, and that's all. They already have the environment, they already have where to feed, and that's just all. And also in terms of the lab, the thing is not as costly as you were breeding mice or rats for practical. So it is very ideal for resource limited settings and also for training students. Now, Drosophila may be ready to fly. It's a fly, but is the environment ready to let it take off? So let's look at the realities. Our Nigerian educational and research ecosystem face some serious challenges, such as inadequate funding. According to UNESCO 2022, Nigeria only invests about 0.35 of its gross domestic product (GDP) in education, against the recommended 4.6%. It doesn't just stop there. It also stated that Nigeria has only 39 researchers per million inhabitants, as compared to the world average of 176. Now, how crazy is that? And we all know funding, money is necessary for effective teaching, for effective research, like for you to be able to develop yourself, or for you to be able to develop others. Funding is necessary, and the lack of it, of course, results in limited hands-on experience for students and researchers alike, which is what we have been experiencing in Nigeria. Another thing is the minimal exposure to practical science. Now, there is this ancient Chinese proverb, which I usually like, is that it says, "What I hear, forget. But what I do, I understand and never forget." Now, this picture of the teacher showing is actually just is just one of the few examples, right example, because at least the students can even see. But there are situations, there are many labs, many biology labs, many research labs in Nigeria that do not even have these to even show the students, not talk of the students doing it themselves. And without the right skills at the earlier exposure, these students lack the skills to take them to the next stage of their careers. Now, another thing is mentorship. Now, how many of us are familiar with the ancient Greek story, Odyssey? Or maybe you've seen the movie? Well, it's the story of Odysseus, king of Ithaca. Yeah, I love that movie. Who, before leaving for the Trojan War, entrusted his son Telemachus to the care of his local friend. That's mentor. His name is Mentor, and that's actually where the word mentor comes from. That is, it's not just a friend, but someone who teaches, guides, and watches over the next generation. So, what exactly is mentorship today? It's the same idea, helping the next generation to find their path. But in many institutions, especially in research, that part isn't always clear. Many young people don't know who to turn to for real career advice. They are navigating alone in a system without structured progression, in a system with few sabbaticals, limited fellowships, and almost no professional development support. Now, this isn't just a gap, it's actually a leak in our pipeline, because we train the talent and we don't see where they go to. Another issue is the brain drain and isolation of trained researchers. We often train brilliant minds only to watch them leave, or worse, we don't have the system to keep them in place, to keep them connected or involved with our national research. Now, according to WHO, the brain drain in Nigeria actually began in the early 90s after the oil boom. And just like Bimbo said, just like she said, is that there are many Nigerians who wish to return to make a difference, but the system isn't giving them the room. Only in extremely few cases, like that of Dr. Mahmoud Bukar Maina, that's director of BioRTC, who is able to get that room to make that difference. So, from chronic underfunding to mentorship gaps to many young Nigerians never getting the chance to experience real science. Those are majority of the gaps. No, it's not exhaustive, but those are majority of the gaps that we are facing. But here's one thing though, our biggest resource isn't just the money, it's our people. It's the talent, the curiosity, the potential. Like, we have brilliant students across the country, and what they often lack is it intelligence? No, but it's the tools, the access, and the guidance. And that's where Drosophila Nigeria comes in. So the big question is, can we really use this fruit fly in Nigeria? Drosophila Nigeria was given birth in the year 2018. It is a homegrown response to some of these challenges that we saw, not all, of course, but some of these challenges. And it began with four like-minded people: myself, Dr. Monsura Bodomasi, Dr. Ahmed Badmos, and Professor Nuhu Muhammad Denjuma, with just one goal in mind: make science practical, affordable, and local. Now, our vision was ambitious though. The initial vision was to train passionate volunteers who would go into schools and work directly with biology teachers, equipping them with the skills to integrate fruit flies into the curriculum using the inspiring inquiry-based materials developed by Professor Andres Prokop. The plan was just to have was to have these volunteers collect feedback from schools, which would then use to adjust our approach and ultimately create Drosophila teaching kit that is tailored specifically to Nigerian curriculum. Reality. Now, we got the people. We still have the people. We still have volunteers, which are now our trainers, Drosophila Nigeria trainers, to volunteer their time. But it didn't feel right to have them volunteer their resources, like their maybe money to go to the schools, money to take lunch at schools and all that. It didn't feel right. So instead of giving up, we changed our strategies and we built by building a pipeline. And this pipeline involved starting with the students, with outreaches, supporting teachers, and enabling researchers. It's not perfect though, but it's working. It has been working, and it will keep improving. And it's inspiring change from the grassroots. So, because when teachers get trained, students get inspired, the fly becomes the gateway to rediscovery. So far, since 2018 that we started, we have been able to introduce 25 schools and spark the interest of 401 students on digital using Drosophila. Yeah, it has increased their engagement. We've made them to see that science isn't abstract, it's alive, it's discoverable, and it's something that they can do. Also, at the same time, we began training and mentoring both undergraduate and postgraduate students to use Drosophila in their research, some of whom even changed their research entirely from the use of mice or rats to Drosophila. And so far, myself, I have supervised about 38 of them, and their work, even at undergraduate level, has received awards and gained attention and recognition at the National Universities Commission team during the departmental accreditation in 2023. Now, this mini lab has provided resources that have helped some of these brilliant researchers to earn their doctorate degree. And one of our most recent cases, my favorite, is Dr. Zainab Muhammad Bello from the Department of Anatomy, who completed her PhD last year, but before then, she had been training, she had students of her own completing their undergraduate degrees in Drosophila. Now, it's not just about women supporting women, believe me, but it's just about the passion I see in her. Seeing her for Drosophila, she's into Drosophila, she's into aging. Yeah, it's the passion and also the deep satisfaction that Drosophila Nigeria's vision is being realized. So we also have trained about 68 biology teachers across Nigeria. We focused on teachers training and helping biology teachers training, to be precise, helping them to understand how they can use fly and why it matters, and how they can use this fly to bring back life into their subject, because biology is life, study of life. So they had to bring it back. Then we've also been able to donate resources such as foldscopes that were donated to us by the ICGB Italy, neurobiology charts that were sponsored by the African Science Literacy Network, and also some writing materials to about 39 secondary schools. Also, in 2017, before we officially began the Drosophila Nigeria activities, I read through the Nigerian biology curriculum and also the modern biology textbook. And using Andrew Pocop's Drosophila Nigeria and Droso for Schools, because that's where it actually came from, Droso for Schools resources, I identified key topics that could be taught more effectively and more engagingly using Drosophila for practicals and to simplify complex biology concepts like genetics, evolution. And in 2020, we wrote a step-by-step guide on how one can easily establish a Drosophila lab and also can be able to handle the flies. Then in 2022, I simplified various experiments together with Andres, various experiments that can be done using Drosophila in our undergraduate and postgraduate researchers, even in resource limited settings like ours. And all of these materials are freely available and downloadable at our website, which is this. Also, these are the states of Nigeria that Drosophila Nigeria has actually made an impact on. Believe me, we are just starting. We are evolving. So we are scaling it up. This is our roadmap to 2025 to that's this year to 2030, God's willing, and to expand to new states, do new projects, and also influence policy and create regional hubs. Then, what have been my insights so far from this? Capacity isn't just built with equipment, it's built with people, relationships, mentorship, all the shapes, and small wins, which creates lasting change. You don't need to travel out, you don't need to travel abroad to start doing meaningful science. We started Drosophila Nigeria in a classroom in Zaria. Demonstration to be precise, that was my secondary school. But we always had a global mindset, adapting international best practices for our local context. Science is global, yeah, but solutions often start local. Another thing that I have also gained from this journey of mine is that teachers are key multipliers. One inspired teacher can influence hundreds of students over time. And that's why we also focused on teachers training, because if you empower the teachers with the right tools, you empower generations of future scientists. Also, international collaborations matter, and so does leadership. Collaborations with groups like Manchester Fly Facility, Just Africa, and Trend in Africa have been very instrumental. They provided the training, the exposure, and the resources that we couldn't have access on our own. But what made the impact sustainable was ensuring that the vision remained rooted in our local context, because true collaboration empowers and our partners have always supported us in taking ownership and leading the change from within. That's the kind of partnership that builds long-term change. Another thing that I have gained from this is that mentorship is as in sustains generational growth. That yeah, equipment is good, but mentorship is better. Students need someone who believes in them, who checks in on them, and who guides them. That's how you build continuity, by lifting the next person as you rise. The spirit of Ubuntu. I live because you live. Also, don't wait for ideal conditions, because believe me, there are no ideal conditions anywhere. You may not have the perfect lab, you may not have enough funding, you may not even be recognized, but start anyway. Start where you are. When we began, we're still using, but at least progressed. We began using this used water Bama bottles. Yeah, used Bama bottles. I remember going to Sapungari market to I don't know what they call these guys, though it's in Hausa. So I got them, took them home, washed them, autoclave them, and then still using. So it was a recycled thing, an innovative thing. So start where you are. Progress doesn't need perfection. It just needs persistence. So please join us as a trainee, as a sponsor, as a mentor, as a partner, as a trainer, as a faculty, as a participant, or simply as an advocate of this model. There is room for everyone in every capacity. The future in Africa must be homegrown with passion, community, and science as our tools, or arts, like the case may be. We can build a generation of researchers who thrive at home or abroad, but who remain deeply connected, actively contributing, and proudly investing in African scientific future. Please join the movement, support the vision, help us scale the impact together. You can see we're just few in numbers. There's room for everyone. Because as we've seen today, why Drosophila is cost effective. It drives high impact research. And in the end, strong science or strong research doesn't require expensive technologies. It just requires continuity, commitment, and a system that grows across generation. So you can scan this. This will take you straight to the Google form in which you can be able to join us, and with what capacity you'd like to join us. There's room for everyone. Arts, sciences, social sciences, history. There is room for everyone. And I would like to sincerely thank my mentors, our founders, our Drosophila Nigeria trainers, and every individual like Professor Darren Obert and organizations that have supported this journey. To every teacher, every student, every researcher who has joined this journey along the way, thank you for believing in the vision. It's not easy to believe in something that was actually new. And to Farah Instal Africa, thank you for giving me the platform to share the story. This is only the beginning, and I'm grateful to be working it together with all of you. And to the audience, I say a very big thank you for your time and attention. And I'll leave you with this quote: if you can think it, Drosophila can help you achieve it. So thank you for your time and attention.
Thank you very much, Ma. We are so grateful. We are delighted to have you in our midst. And this section has been an impactful one. In fact, your story has really, really impacted me. Thank you so much, Ma. And I know it has impacted others that are listening to it. Yes. you can start using Drosophila even from anywhere. Wow,
Anywhere.
that is such a yes, it's such a great one. Thank you so
Thank you.
much, ma'am. Yes,
Thank
ma'am.
you.
Uh, I don't know if we have any question from the presentation. If we have any question, you can drop it in the chat box. She will help us answer them. If you have any question, be it on YouTube, you can drop them in the chat. Ma, before they drop the their questions, I have a question here, ma. Ma, you made mention of using of jar, like Bama jars,
Yes.
mayonnaise jars. Okay. Ma, can you explain how we can go about that? Can you expatiate on it, on how we can go about the use of
Oh,
jars?
okay. Yes. Like I said in my presentation, that all these how you can see that from even the cutting of foam plugs, the jars are used Bama bottles. 1 minute. Let me just get something to show you.
Okay, ma. Okay, ma.
Just a minute. Yeah. So this something like this. This is honey in it, but in Nigeria we have bama in it. So what we do is after using the honey or using the bama, we get the used bottles from the market. There is a place where they usually sell all those used bottles. So we got it, and of course you uncap it. You wouldn't use the cap because it's not porous. There won't be airflow. So we use after washing, we use it, and then we make a cap on our own using foam plug. That's what we use to make cushion. What we use to make cushion, that's what we use because it's porous enough, and then there can be exchange of air between the flies and the environment. So that's how we use it.
Okay, ma. Thank you, ma. And then, which part of the insect, which part of Drosophila do we use when we do our research? Like if we are using rats now, we extract some organs in the body, and then we carry out our research there. Then in the drones, I don't know what are the things, what are the parts we use there.
Every part, every life cycle stages. Just like you said, you harvest organs in rats, you can also harvest organs in Drosophila, mimicking human kidney, just such as the tubules in Drosophila. We have the liver, such as the fat bodies. So you can also harvest those organs, but that's after you've exposed them to treatment. You can use the whole fly. You can also use their embryo stages or the eggs. You can use that. Okay, I even remember using that during my research in Germany, where I was trying to check the development, the mitosis I was taking on, and how the ventral nerve cord usually develop. So I use the embryo stages. Then you can also use the larva stages for different assays, including behavioral assays. And you can also use the pupa or pre-pupa stages also for different kinds of research. And finally, you can use the adults in various ways. Yeah. So all stages, every part is usable in Drosophila research.
Thank you so much, Ma. Thank you, Ma. Our people are very glad to have you, and they have been inspired by what you've discussed with us today. And they're asking for your contact, yes, your contact, your phone number, and email. Okay, a response has been given on the YouTube. A response has been given already, so you might see some of us coming to you to ask some question, maybe personally.
Yeah, and I'm seeing something from Fire. I'm seeing, oh, is that usual stuff? I can see an email, rashidat.abu@fire. Is that is that it? Hello?
Okay, yes, Ma, you were tagged.
Okay. Okay,
Okay,
yes.
yes, you were tagged to the question that was asked about your contact and and so that's your, I think that's your mail. So that's how they can reach out to you.
Okay. And
Okay,
yes,
so
and they can also reach out to the forms that I see, Google form that I see, and also through the website that is about yeah, that's npds drosophila at gmail.com. They can also reach me there. Yeah. So
okay, ma. Thank you so much, ma. We are still awaiting questions. Please, if you have any question, just drop it on the chat box. Drop it on the chat box. Some things you've been curious about, with the use of Drosophila, you can drop them on the chat box. Okay, someone just dropped a question now on YouTube. It was like, what material are used as those insects are small to touch? Like, what materials are used as those insects are small to touch? Or what material do you use since they are smaller than other organisms that we handle in lab?
yes, there you use several materials, starting with a microscope, but they are not that small compared to microbes. You can, well, for some assays, we just use our eyes. Like when you are talking about behavioral assay, locomotion assay, we use our eyes because you can see them. But if you are talking about maybe dissection or wanting to get one part of the fly or wanting to see maybe the peristaltic movement of the larva, you use a microscope to do that. So it's actually quite handy. Yeah. So those are the stuff that you use, just a microscope or as simple as a magnifying lens. That's for a classroom. You can use a magnifying lens if you just want to see the peristaltic movement. But other than that, your naked eyes can be used and also your hands to manipulate the flies. Yeah.
Okay, ma. A question from my own hand. I would like to ask that can it be used in genetical studies, like in the aspect of genetical engineering, genetical modification, and all? And how can we do that? Can we manipulate their gene? Are they easy to manipulate, ma'am?
Oh my God, I forgot to mention that Drosophila, especially Drosophila melanogaster, is nicknamed Cinderella of genetics. It is the Cinderella of genetics. It's the icon of genetics. So you can use it. They have just four pairs of chromosomes, and that has given them the power. That has given you, the researcher, the power to be able to manipulate them anyhow you want. You can easily create mutants, you can easily create transgenic lines. Yes, they are the perfect tool for genetic manipulation of any kind. Yes, so you can.
Thank you so much, ma'am.
Yeah.
Thank you so much, ma'am. I'm still waiting for questions.
Okay.
Yeah, there are many remarks on the YouTube page.
Yeah.
Thank you very much, Ma, for the insightful and wonderful presentation.
Thank
Thank you
you.
so much, the presenter and the organizers. Wow.
Yeah. Thank
There
you.
are many remarks, Ma. Thank you, Ma. I must say, the this is so inspiring. So I would like to tell everyone viewing us now that a certificate will be provided after the meeting. So you have to go through the link that you see on your screen. There's a link on the YouTube page. You go through the link to get your certificate. So certificates will be available. So go through the link. You can scan the link on your page too to get the certificate after the meeting. So I would like to bring to our notice the coming events. There will be other events, other inspiring ones, other insightful ones this August and in coming months. So we should do well to look out for them. So while I'm still doing that, I'm still looking out for questions.
Okay.
Still looking out for questions. Okay. In the absence of question, Ma, I must say thank you, Ma, for today. Thank you for your time. Thank you for sharing from your wealth of knowledge to us. We are so grateful, Ma. And I know that well, if we invite you next time, you will gladly honor our invitation. So it's a privilege. It's a privilege having you here, Ma. Thank you, Ma.
It's all mine.
We are grateful.
Thank you.
Yes, Ma. Also, the following information is for us. Let's watch out for the next webinar, which will be coming up this Thursday, 28th of August. There will be a webinar on descriptive statistics and data visualization. Let's mark it on our calendar. It's going to be an impactful and practical one. There's going to be a practical section, so let's make sure to mark it on our calendar and be available. And also, let's watch out for the next series. This is the 14th series. Let's watch out for the 15th series. And I know it's going to be greater. Also, there is MNCH, that's mother care. There's MNCH webinar also, which will be coming up very soon. Let's watch out. The flyers will be shared, and when the time comes, we will know. So let's just mark them on our calendar once the date is out. Thank you so much, everyone, for attending today's webinar. I'm so glad having you here. We are so glad at Firat having you here. To the organizers and the teams behind the scene, I say thank you for being part of the webinar, and also to everyone who attended and participated in today's webinar. Thank you for being part also. You are recognized, and your presence made it special. It's a special one because you are present. Thank you so much. And lastly, once again, I would like to thank our speaker once again. Thank you so much, Ma, for your time, for your knowledge. We love you so much, and we love to have you some other times, Ma. Thank you so much, Dr. Rashidatu Abdulazeez. Thank you, Ma.
So much.
I am, yes, Ma.
Yeah, thank you so much, Rebecca. Thank you.
Yes, Ma. So this brings us to the end of the meeting. Thank you all.